this has a squiggly line here, which means that it can be alpha or beta. So this compound here, this is maltose. 2 glucose units linked together with an alpha 1,4 linkage is maltose. Okay, and when you say it's maltose, it's not enough to say just maltose. Well, this one is just maltose because we didn't draw this in any configuration. But it's going to be in one or the other. So if this one is, this hydroxyl group is in the down position, then this sugar here is alpha maltose. If this hydroxyl group is up, then we would have beta maltose. So even though, and maltose, by definition, this one is alpha, always. So if we have a beta maltose, it's referring to this hydroxyl group. It doesn't mean that this one's beta. This one's always alpha. If this one's beta, then it's cellobiose, different sugar altogether. Let me see, is there, I don't think there's anything else I wanted to say on here, but I'm feeling like I'm missing something. Well, we'll come back to it if I remember. Oh, that's what I wanted to say. So this bond right here, you see how it has this 90-degree angle? So typically in organic chemistry, if we have that 90-degree angle there, that would mean that there's a carbon right here, right? But in this case, that's not what it means. So this carbon right here is linked directly to this oxygen. And we just draw it like this to exaggerate that it's in this down position. It's not actually, we didn't add a carbon here. So be really careful and you know, understand that this is just the way that these sugar, you know, molecules are typically drawn, is that you have this bend and you would understand that this, we didn't pick up an extra carbon. So this is directly linked here to this oxygen. Here's 2 disaccharides that we see all the time. At the top we have lactose. And lactose has this strange bond here. So again, this bend right here does not mean that there's another carbon. This carbon is directly linked to this oxygen, but we want to exaggerate that this is in the up position and this is in the down position. So here is the name of lactose. And so you name these by starting at the non-reducing end, this side, the sugar that can't open up anymore. So this is galactose. Galactose, remember, is the C4 epimer of glucose, so it differs only at carbon 4, which is this one right here. Let me get this thing so it stops disappearing. This one right here. So instead of being down like in glucose, see this down in glucose, it's in the up position. So this is galactose. And galactose has its anomeric hydroxyl group in the up position, so it's beta. And it's a 6-membered ring, so it's a pyranose. So it's beta D-galactopyranosyl, and then the linkage here is 1,4 again. So this 1, 2, 3, 4 to the glucose unit. Also in the pyranose form. And we haven't drawn this hydroxyl group in any particular configuration. So this is just lactose. But we could draw this down, and then it would be alpha lactose, even though this one is beta. If we drew this down, it would be alpha lactose. If we drew it up, it would be beta lactose. You can shorten everything this way. So the abbreviations for all these sugars are just the first 3 letters, with the exception of glucose, which is GLC. We abbreviate glucose GLC instead of GLU, so we don't get it mixed up with glutamate amino acid. Next, we have sucrose. Sucrose is a little bit different than these other 2 disaccharides that we've looked at. So I drew them on top of each other because it's easier. Because these sugars are both linked at their anomeric carbon. So the linkage here is a 1-2 linkage. This is fructose down here. And fructose, remember, has a ketone group. So its reactive group, the ketone, is on carbon 2. So when we name sucrose, we have no reducing end. And we have no non-reducing end. They're both non-reducing. So we don't know where to start naming. So we just name the sugars in alphabetical order. So F comes before G, so we put the fructose first. So this is the fructose, and this fructose, this is the first time we've seen a 5-membered ring. But this is what it looks like. And remember, that's a furanose. I have this here. This is fructofuranosyl group here. So we know that it's in a 5-membered ring. And that is linked at the 2 position where its ketone was to the 1 position on glucose. This 1 position. Fructose, this hydroxyl group is up, so it's beta. And this hydroxyl group is down, so it's alpha. So this is beta fructose and alpha glucose. And then here's how we would abbreviate it. So because both of these sugars are linked at their anomeric carbons, that's why sucrose is a non-reducing sugar. So it does not participate in non-enzymatic browning. The only way that we can get sucrose to participate in non-enzymatic browning is if we hydrolyze this linkage and have glucose and fructose. And then it will brown. But by itself, without breaking down, sucrose will not brown like these other reducing sugars do. Okay, let's move on to some maybe more fun topics. So we're done talking about structures. Now let's talk about functionality. So we have lots of things that sugars do in foods. Down here, we have some different factors. Concentration, the type of the sugar, and so on. And all of these factors are going to influence the functionality of sugar. So if you change the pH, the sugar functionality may be different. Or if you change the matrix, the functionality of sugar may change. Here are the functional properties of sugar at the top. Of course, sweetness, but there's many other important functions that sugars play in our foods. So we've already talked about the sweetness property of sugars, so I'm not going to go over it again. But you remember that we have that sweet taste receptor that's a G protein coupled receptor. that will bind to sweet molecules and then release those G proteins into the cell and then release cytokines so that are neurotransmitters that we identify as sweet. And then we also already talked about the Schallenberger and Acree theory of sweetness and how a sweet molecule needs to have an electron donor, an electron acceptor region, and then also a nonpolar region at particular distances from each other. So I won't go over that in any more detail. I think we talked about that already, but that's the property of sweetness. So let's go through a little bit more about sweetness and some of these different sugars. So in this graph, we're looking at the concentration of several different sugars and how sweet they seem to be. So of course, as we increase the concentration of a sugar, the sweetness is going to go up, right? But what do you notice here? These aren't straight lines, right? Some of them are curved in a concave kind of way and curved up like this, and some curved down, convex kind of curve. So if we have well, let's look at sucrose first. The sucrose is purple. So sucrose, what we notice is that at these low concentrations, we get a rapid increase in sweetness. But as we get the concentration higher, the higher and higher that concentration gets, the less and less impact that causes on the sweetness. And up to a point where no matter how far we increase the sugar concentration more, the sweetness doesn't really increase anymore. So this is called sweetness compression. So if we reach an asymptote, a portion of the curve where we don't get any more sweetness for the addition of more sugar, that would be sweetness compression. So that is characteristic of sucrose and fructose. And then these other sugars down here, glucose, lactose, maltose, and trehalose, those ones have a different shaped curve. You see here at low concentrations, you don't get much increase in sweetness. But as that concentration gets higher and higher, the sweetness intensity comes out more and more. And so that is called sweetness expansion. Because as we increase that concentration, we get more impact for every addition of that sugar. So that sweetness expands. So one thing that you also want to notice from this graph is the relative sweetness between these different sugars. That's probably, the real important take-home message. So if you were to rank these sugars, fructose is the most sweet because it's the highest on this graph. And then sucrose, and then glucose, and then these other sugars, including lactose, that aren't very sweet. So that's another thing that you want to pick up from this graph. The relative sweetness of these different sugars. And then what we talked about at first, sweetness compression, these two sugars at the top, and then sweetness expansion, those sugars that have expanding kind of curves. Next, we have persistence of sweet taste. So this is showing at different concentrations of sugar how long that perception of sweetness stays in the mouth after you've swallowed that sugar solution. Ideally, we want that sugar, that sweet taste, to go away quickly. So we want to drink our soda or we want to eat our candy, and it should be sweet when it's in our mouth. And then when we swallow it, which shouldn't be sweet anymore. And that's a property that sucrose has. Sucrose, again, here is in purple. And you see that this line is pretty flat and fairly low on this curve, right? The persistence of sweetness is very quick. So, less than 10 seconds, that sweetness is generally washed away from our mouth. Some of these other sugars, like maltose, lactose, and trehalose, those sugars that are not actually very sweet, that sweetness, though, sticks around in the mouth for a long time. And so that is actually not desirable generally. So it makes these sugars difficult to use because not only do we have to use higher concentrations to sweeten things, but that sweetness persists, which kind of leaves this lingering sweetness that's undesirable. This is a major problem with artificial sweeteners. So we don't have any artificial sweeteners on this graph, but if we did, those artificial sweeteners generally have very long sweetness intensity, persistence of sweet taste. So you would be in the minutes instead of the seconds. And so that is one major limitation of a lot of these artificial sweeteners, is that their persistence of sweetness lasts for a long time. Some of those protein sweeteners can be hours even. Okay, so texture is another property that sugars impart to foods that's very important. So they provide texture to beverages. So we would usually call that not texture, but body. So they provide body to beverages. And you can immediately notice that if you taste the difference between a sugar-free and a sugar-containing beverage. That sugar-free beverage will always seem like it's missing something. As it goes in your mouth, as it coats your mouth, it's missing something. And that thing that it's missing is the viscosity of sugar. And that would be, that sort of body that it imparts. And you see here that at cold temperatures, here's 5 degrees, just above refrigerator or at refrigerator temperature, these sugars at 10% increase the viscosity a little bit. So you see that increase is not actually very much at all, but it's enough for us to perceive quite a difference in the mouthfeel of these beverages. And then if you look here, this is 40% sugar. This is like a syrup that you would use for canning peaches or pears or something, a heavy syrup. Quite sweet, but you see that viscosity is now basically 10 times higher than water itself. So at those higher concentrations, it has a drastic effect. This one's more reasonable for beverages. Soda, carbonated beverages would be close to this 10%, maybe a little bit higher. Juices are going to be higher, 14, 16%, something like that. So that's just for comparison. In baked goods, they have a really important role in texture. So first we'll talk about this picture here. So this is 2 cookies, chocolate chip, one made with all sucrose and one made with just a little bit of corn syrup added. So you can immediately notice a difference here. So sucrose, one thing that it does is it tends to cause these surface cracks. And that's because the cracks are caused because that surface dries out before the cookie is actually done spreading. And so then you get all these cracks. And that's because sucrose is so the hygroscopicity, remember that term, the ability for sugars to take water from the atmosphere. So the sucrose doesn't take very much water from the atmosphere. So it's usually pretty dry. And so that surface tends to remain dry and crack. And there are other sugars, like those that are in corn syrup. You know, I don't know. This is either just corn syrup or maybe high fructose corn syrup. So we either added just glucose or glucose and fructose. But at any case, both of those sugars are more hygroscopic than sucrose. And so they will pick up moisture from the atmosphere, and they will hold on to moisture. And so they will keep that cookie from drying out so much. So not only do you see on that cookie on the top that it doesn't have those cracks because it didn't dry out before it was done spreading, but it also is going to be softer. So the cookie on the top is going to be chewy. And because those sugars have held onto that water. The cookie on the bottom is going to be crunchy or crispy because that sucrose is dry. So I want to list some other functional properties of sugars. So one is that they provide bulk. So that's, maybe it seems like that's not so important, but it turns out that it's quite important when you add so much sugar to these products. So like in these cookies here, you're adding a large amount of sugar. And so that's a large amount of solids to this product. And if you took all that out, say, and replaced it with an artificial sweetener, you would take out all those solids, and the properties of that product would completely change. So it's really important that bulk is provided. So making sugar-free cookies, for instance, you can use sugar alcohols, if you want it to be sugar-free, and still provide that bulk. But using a straight artificial sweetener would not work. You would end up with something that was like kind of like a sweet cracker or something. It would be very different from a cookie. All right. Next, we have into batter or dough. So sucrose or sugars allow air to be incorporated into a batter or a dough. And that's going to be very important for that product to be leavened enough. So if you remember in our additives section, we talked about baking powders and baking soda. And those are there so that we can create CO2, which allows air bubbles to expand when that product is in the oven. In order for us to get even more air into the batter before it goes in the oven, sucrose does that, together with fat. So fat and sucrose are both going to be combined together and beaten together, and the combination of those two things, that beating, gets air into the batter so that we get all the leavening we can. Because an important thing to remember is that, as that water turns into steam in your dough or batter inside the oven, you're going to lose all that steam to the atmosphere unless you've got little air bubbles or little carbon dioxide bubbles in the dough that can capture that steam and help it expand. So really important property, especially for cakes. It's important in cookies, but especially cakes and stuff. It's extremely important. There are actually a lot of cakes that rely entirely on this for leavening. Okay, prevent gluten formation. What's gluten? A protein, good. Wheat protein. So if we're using a wheat dough and we mix that with water and knead that or mix it somehow, then we will get gluten formation. So gluten formation is really important for breads. That's what makes that dough elastic so that it can trap gas during leavening, during yeast activity. But in products like cookies and in products like cakes, we don't want that because it will make them tough. And sugar is going to help to prevent that from happening. And sugar does that by trapping all the water. So any water in that batter or dough, the sucrose is going to hang on to that. And it's not going to give it to the gluten or the protein to be able to hydrate and form that gluten. So of course, these batters and doughs are also, that gluten is inhibited by the addition of fat as well. But sugar plays an important role also, and right now we're talking about sugar. So next, it delays starch gelatinization and also protein denaturation. So I just mentioned sugar holds on to all that water. And when it holds on to that water, it's going to make it so that it's not available for protein and it's not available for the starch. And water is important for proteins to denature or for starch to gelatinize. So if we've yanked all the water away from those materials, then they won't be able to gelatinize or to denature until later, until the temperature gets higher than normal. So why is that important? It's going to allow that product viscosity to stay low so it can expand to its maximum expansion before the starch gelatinizes and the protein denatures, which is the point when the structure is set and it's not going to expand anymore. And so if it sets too early, then we're not going to get as much expansion as we normally could. And so sugar is going to help to delay that so that that expansion can be as much as possible. If we have expansion occurring, but the viscosity is too low, then we can still lose all that gas, right? Because we don't have enough structure to hold on to it. So sugar is also going to provide viscosity. So that's what's going to help to keep that viscosity high enough to trap gas before the starch and the protein gelatinize. And in these products like cakes and cookies, a lot of times the batter, it has so much sugar in it, a lot of it's not even dissolved yet. But once it gets in the oven and starts heating up, it will dissolve and create that viscosity that you need to keep the gas there in the batter or the dough. Okay. Next, let's talk about fermentation. So first we'll talk about yeast. And here we're talking about baker's yeast or brewer's yeast, which are the same genus and species, Saccharomyces cerevisiae. So this type of yeast here, these are the sugars that it can ferment: glucose, fructose, sucrose, and galactose, and also maltose. So kind of a limited range of sugars that it can use. But, and I put here galactose in parentheses because we don't often see free galactose in foods. It's typically present as lactose, which you do not see on this list. So adding, say, dry milk or milk to a dough, yeast leavened dough, is not going to provide sugars for the yeast to metabolize. Here we have gas production. So this is in a bread dough, and we have different sugar concentrations, and this is how much gas is produced. And so you notice that sugar, it's an important food for yeast. But you see that if you get too much of it, that gas production starts to decrease. And this is really just because of osmosis. So as we get up there sort of above 8% sucrose in the formula, there's sufficient osmotic pressure that the yeast has a hard time. So much dissolved solids in the aqueous phase outside the cell that it's tough on the cell. It starts to dehydrate the cell. And so, so that activity goes down, gas production goes down. Getting sucrose concentrations up this high is not at all uncommon. So there are a lot of baked goods that are yeast leavened that have 15, 20, 25% sugar in the dough. And so this really happens a lot. And you'll notice it as if you make a dough that has a lot of that sugar in it, that it will take sometimes even twice as long to rise. And it's also typically compensated for by adding more yeast. So you'll add more yeast and give it longer in order for the doughs to develop all the way. Sandwich breads and stuff are typically going to be down here, somewhere between 5 and 8%, so that we have a lot of sugar there for the yeast to metabolize, but not so much that the metabolism is slowed down. There's also another case that's important for fermentation. This is making yogurt. So these are yogurt bacteria here, Lactobacillus delbrueckii bulgaricus, and then Strep thermophilus. So if you look on your yogurt container, typically you'll see both of these listed as active cultures. And these two bacteria kind of work together. So the lactobacillus can metabolize lactose. So that's different from the yeast. So it takes in lactose and hydrolyzes it to its constituent sugars. And then for some strange reason, it spits the galactose out. And it only uses the glucose. And goes through glycolysis, does its metabolism. So that's where the strep comes in, is because the strep can use galactose. So it's perfectly happy to be around these lactobacilli to get this galactose from them. So it can take galactose in, and it can either just metabolize it a couple of different ways to glucose, or can, this right here should say glucose. I apologize for that. So if you isomerize galactose to glucose, and then go through metabolism. So they work together there, and this isn't the only thing that they do. Let's see, the lactobacillus here also is going to produce some amino acids that are important for the strep to use, and the strep is going to produce some vitamins that are important for the lactobacillus. So they work together in other ways besides just this. But I think it's interesting that the lactobacillus takes in this lactose and just uses the glucose almost so it can help out that strep to live with it. So one thing that you don't see here is sucrose. And for most of these yogurts that we eat, there's a lot of sucrose in them. Something between 10 and 20% sucrose is added to these yogurts, quite a lot. And sucrose inhibits the growth of these microorganisms. So when making yogurt, you would allow the yogurt to set, and then once it has set, then you put all the sugar in, once these bacteria have done their job. So even though, yeah, that's important to remember. All right, preservation. Sucrose, historically has been used for preservation of foods, just like salt. And so on this graph, I just have the change in water activity as the sucrose concentration increases. And so you see you have to get a pretty high sucrose concentration to have much decrease in water activity. So if we're talking about inhibiting bacteria, we're up at 50% sucrose, which is almost too sweet for most products. In a product like a jam or a jelly, those are going to be typically here maybe 65, close to 70% sucrose. So we're not going to get much growing in those because we're way out here. So that's an example of really just taking advantage of that decrease in water activity that sucrose provides to prevent stuff from growing in it. Okay, let's go into more detail about non-enzymatic browning. So we've already talked about this a little bit. And now that we have a little bit more understanding of sugars, though, we'll go into it in a little more detail. So this is just an overview. We have two types of non-enzymatic browning that are important to keep separate from each other and understand the differences. So we have caramelization, which involves only sugars and occurs only at very high temperatures. So substantially above boiling water temperatures. This is where we get caramelization to occur. We have a lot of dehydration that occurs, and then pyrolysis. And those pyrolytic products can be reactive and form brown colors. And they can also be aroma compounds. We have the Maillard reaction, which involves sugars and amino acids. And it will occur at hot temperatures, but not really hot. And we can form the same kinds of compounds with the addition of nitrogen from the amino acids and form brown colors, flavors, and aromas. We also can form acrylamide in the Maillard reaction. And acrylamide is a carcinogenic compound. It's a toxic compound. We don't want it. And so we would want to take steps to avoid acrylamide formation from this browning reaction. So we've seen this already. Here is our sugar, and I'm just showing the top of our sugar because that's the reactive part. And here's our amine from the amino acid, and they're going to condense to form this Amadori product. And if you remember, this Amadori product is the point at which non-enzymatic browning is going to go forward. Up until then, it could go backwards. And then here again, this is the formation of a dicarboxy group here. In this case, an aldehyde and a ketone. And this would be reactive in Strecker degradation, and then as well as formation of these furan or furfural derivatives. Because these diketo groups here are so reactive, they can break here in between them and form smaller molecules that give us the aromas from the Maillard reaction. Here's Strecker degradation again. The diketo group with an amine. They trade places, and we form that aldehyde with one less carbon than before. Here is acrylamide formation. We have some amino acid and some dicarbonyl compound. So something from the Maillard reaction. And we go through all these steps here and pass through this Strecker degradation reaction and form eventually acrylamide. This amino acid here can be one of several, but the majority of acrylamide that is produced in food would be from asparagine, the amino acid asparagine. So this reaction really only occurs in foods that have a lot of asparagine in them. So asparagine gets its name from asparagus, which is where it was discovered. So asparagus has a lot of asparagine in it. But we typically don't eat a ton of asparagus. And when we do, it's often boiled. And so there's no Maillard browning. So asparagus, despite having a lot of asparagine, doesn't necessarily give us a lot of acrylamide. Instead, we have other foods that we eat a lot of that give us this compound. So one would be potatoes. Potatoes have a lot of free asparagine. And there are many ways that we cook potatoes that result in non-enzymatic browning. So for instance, potato chips or French fries. And so when these products are consumed, there would be a significant quantity of acrylamide. And there are lots of steps that have been taken to mitigate this. So trying to remove the free asparagine, for instance, by soaking the material before it's fried, or by adding, say, asparaginase, an enzyme to get rid of it. You can also try and get rid of the sugars different ways. So there are steps that have been taken to try and minimize that. Another very important source of acrylamide would be bread products. And that is because we eat so much bread, and wheat has some free asparagine in it. And so wheat products are going to contribute some to our intake of acrylamide. And then coffee would be the last one. So if you're a coffee drinker, if you drink a lot of coffee, that's going to have tons of acrylamide in it through the roasting process. So those three things, fried potatoes, bread products, especially bread products that are brown. So, if you see a lot of browning on the crust, or if you make toast or something, those are going to have more acrylamide. And then coffee has a lot from the roasting process. And if it's a dark roast or something that's extensively roasted, it's going to have more, of course. There's been recently a lot of research on this, and whether or not it's actually dangerous is still up in the air. I think the intake is still pretty low, but acrylamide is quite toxic, so it's something we do want to avoid as much as possible. Okay, we're out of time, so thanks everyone. We will have our review on Friday and talk more about this. So.